Good morning to all again. Myself, Pankaj Yadav. I am an assistant professor at Department of Bioscience and Bioengineering at IIT Jodhpur. So, welcome to this session of the fundamentals of biology session. So, this is the session on big data in biology. So, today I'm going to talk about some basics in big data and also in the coming sessions on talks from different speakers across the world, you will listen to the more applications and also some analysis in this field. So the objective of this workshop in this session is to introduce you to the basics of this big data field in biology. So let's start. Okay, so before we go further, let's start with some definitions. So the first thing is probably in your mind is what is data. So let's start with very basics. So data is basically a set of values of qualitative or quantitative variables about one or more persons or objects. So how we define this is actually very simple and straightforward. So we can have different kinds of variables. They can be mostly can be categorized in qualitative or quantitative variables, and how we do the observations on these variables on different persons and objects. That's called the data. And now the next thing is the data set. So its data set is a collection of related sets of information composed of separate elements that can be manipulated computationally as a unit. So basically, it's a collection. It's a collection of related sets. So, for example, it can be a collection of height and weight of certain set of individuals. So, another definition is that is very important nowadays also in data science is database or data repository. It's a virtual data storage that stores, organizes, validates, and makes accessible core data related to a particular system or systems. So we will also see some examples of the databases in this coming workshop sessions. And so another definition is metadata. That is sometimes also confusing to some of the researchers and students is a metadata. So it's actually a data that describes your data. For example, it can, if you consider in terms of very basic example. So let's say we have a publication. So in a publication, we can have metadata as a title, authors' names, abstracts, keywords. These are all your metadata that describes your publication, right? So this is metadata, and we have also next is workflow. So workflow basically defines a series of tasks for processing your data. So there are different workflows are very important when it comes to data analysis. So you have to make some workflows to create or to do some tasks. For example, in NGS, in next generation sequencing data analysis, you will have a set of tasks that need to be completed before you get some to conclusion. So workflows are very important in data science. And now coming to the most important definition, I think, for this conference is data science. What is data science? You will also hear a lot of these different definitions on data science from different speakers, from different presenters in this conference. So to me, the data science is an interdisciplinary field. It requires skills of several basic skills, at least in computer science, mathematics, and biology. When it comes to domain knowledge. So interdisciplinary field in which quantitative and analytical approaches, processes, and systems are developed and used to extract knowledge and insights from increasingly large and or complex sets of data. And also Nowadays, deep learning has also become an important methods to that are kind of um, uh, that is complementing data science field. It's deep learning. Deep learning, in a very basic definition, is that it's a type of network, and I'm not going much into technical details of this. It's a type of network in which different successive layers uses output from previous layers. So it's a network of layers. Each layer uses information from a previous layer. It's similar to, you can think of it as a communication pattern in a biological network, a nervous system, if you come from a biology background. So another last definition that is more relevant for this session is omics. Omics in the sense of multi-omics. Omics is a collective term again. It's a collective organization, characterization, and measurement of biological molecules that translates into structure, function, dynamics of an organism or organisms. Examples are genomics, proteomics, metabolomics, and other sources of biological data which ends with the omics. So we will also hear more on this in the coming slides. Okay, so what are biological data types? And I think from this conference point of view, it is more important to introduce you with the different data types that are more popular or that are more relevant in biology. So genomics is one of the probably the most basic types of data types in biology. So what does it comprises? It comprises of nucleotide genome sequences, metagenomic sequences, and what can we do with this? We can do several things with this. So we can do gene finding, we can do functional annotation of the genes, we can do sequence alignment, we can do homology determination, comparative analysis, we can also do phylogenetic inferencing with the help of nucleotide sequences. We can also do association analysis, like if you heard about genome-wide association study, GWAS, this is also relevant in that context. And mutation functional prediction of the genes, and we can also do a species distribution analysis to do as people do in the population genetics field. So the next data type is transcriptomics. RNA expression levels and transcription factor binding, chromatin structure information. These are all different data that comes under transcriptomics. So what can we do with such kind of data is we can do differential analysis. We can do clustering, we can do functional enrichment analysis. We can also do transcriptional regulation and causal reasoning analysis. And the next category of the omics data is proteomics. It basically has data on protein levels, also protein structures and protein interactions, how different proteins interact with each other. So what can we do with such kind of data? So we can do we can do protein identification, which protein is actually there in the data. We can do protein functional predictions, what kind of function is done by a particular protein. We can do structural predictions based on the sequence of the protein. We can also do structural comparison, molecular dynamic simulations to study the more logical biological in a biological environment, how different proteins behave in a cellular system. We can also do a mutational function prediction and docking predictions, and also we can do a protein network analysis. So, and the next category of the omics data is metabolomics. It has metabolite and small molecules that are present in our body. And what can we do with the metabolics is we can construct pathways, how different metabolites are involved in different biological pathways, and also different network analysis based on different different types of data, like you can integrate genomics and metabolomics data together. Some slides will come up some follow-up slides will come on this in a few minutes. So another different source of nowadays very important in the biological field is bioimaging data. It's basically comprises of microscopy images, MRI images, CT scans. So these are also nowadays included in the biological big data. Actually, it is one of the important part of the big data in biology. So what can we do with this kind of data is we can do feature extraction, we can do high content screening, we can do predictions of different, we can do diagnosis of different diseases with the help of cellular imaging data, or we can also determine cellular structures, substructures based on the imaging data. And another sort of data is cytometry. It has cell levels, cell phenotypes, how these cells are behaving, or cell population clustering. And this is one of the nowadays also used in single cell analysis. And just to give you some background also in systems biology, it's a huge field. It actually comprises all of the above data sets, data types that I have described. In addition to this, we can also do network analysis. We can also do reversal, causal reasoning, drug target predictions. These are all some subdomains of system biology. Okay, so when it comes to the volume of the big data, so let's say how big comes when we measure it on a scale of the bytes, comparative on scale, then as computer scientists, some of you might already know that the byte is the basic unit of measurement. After that, we can have kilobytes, megabytes, and up to yottabytes. So biological data is basically so huge that in coming times, we will need to go to those upper end scales in order to store and to analyze such kind of data. So you can imagine how challenging it will be when it comes to analyzing such huge amount of data. So, so just to give you a feel and what are different, if you go like more in depth of data kind in data levels in biological research. So we have different types of data that, let's say this is just an illustration that we get from mice. For example, we can have experimental metadata, which kind of mice was used in the experiment, what kind of cell culturing was used, and then we can have different sequencing methods. We will also discuss in a few minutes. What are different sequencing methods? Then from sequencing method, from experimental data, after sequencing and all, we get to the primary data that is probably more relevant for computer scientists and also for data statisticians. So after the sequencing, after this stage, we go to this numeric data, and basically this is where the job of statistician starts, where we analyze primary data, we do pre-processing, we do normalization, different kind of steps to make sense of the data. After doing all, we get to the derived data from the primary data, and from derived data, we go to the interpreted data and knowledge, where we do a lot of data analysis, statistical analysis, do pathway and network analysis. We comes to the knowledge that comes from our experimental data. So this is a basic cycle of data levels in biological research. So you see there are different peoples that are involved in such kind of from here to here. If you think of doing an experiment on mice and creating such kind of knowledge, it's such a interdisciplinary field that different peoples of different skills are involved in this cycle. You can say molecular biologist, probably cell biologist starts here, and you can see the data scientists and computer scientists probably lead us to there. Bioinformaticians and all they work in these different stages of this data levels. So coming to the NCBI, probably most of you just know about NCBI. So this is one of the probably the most important portals that are available relevant in the field of biology. It's from National Library of Medicine. So it's this picture actually just shows you, this figure shows you that how big the data, or how the data has grown over years of time. So if you look at here, so after, like, before the Human Genome Project in 2003 or some prior to that, the data didn't grow much. But after that, just after this Human Genome Project started in around 2099, sorry, and you can see that the exponential growth of this, the amount of data that has compiled. So on this side of this figure, we have sequences in millions that have been available in this NCBI data portal so far. And on the here on the right side, we have users. So you can see that as the amount of data has grown, also the number of people that have shown interest working in this all have also gone up to 5 millions and even more. So this is actually showing us the level at which we have reached, or in these 2 decades, in last 2 decades, when it comes to the biological data. So I just want to show you some example on data science, interests of data science in this field. So NIH has a strategic plan for data science. So I really like this slide, where they have shown this different plans that need to be accomplished when it comes to the data science field. So we have data structures, which in this, basically, their plan is to optimize data storage and security. These are very fundamentals of data storage, and connecting NIH data systems. So different types of data, how they connect to this, and then modernizing this data ecosystem is one of the another plan they have proposed. Modernizing the data repositories ecosystems and support the storage and sharing of individual data sets. Better integrate clinical and observational data into biomedical data science. And data management is another, and analytics and tools. So this is also an important part of their plan to support the useful, generalizable, and accessible tools and workflows for analyzing and for making sense of the data that they have tried to gather, and broaden utility and access to the specialized tools for the public uses, and improve the discovery and cataloging resources. Workforce development is also an important part of this, because without that we will not be able to, what we will be doing with the data we don't know. And hence NIS data science workforce, expand the national research workforce, engage the broader community of the scientists to get involved in data science and biology field, and stewardship and sustainability, develop policies for a fair data ecosystem. So there is also a policy of fair data access and fair data usage and privacy law in the part of their plan. Enhancing this kind of stewardship is also their plan. So this is an example of one example of the big data in biology. So NIH has come up with this big data knowledge, big B2B big data to knowledge. It's an example that has been proposed by NIH. So we have more than, in this BD2K achievements, they have around more than 30,000 individuals have been trained in biomedical data science, and now more than 255 biomedical data scientists educational resources are available as a part of this project. And integrative digital media for analyzing biomedical data has also been is available now as part of BD2K. And now more than 200 software and tools are also available as a result of this project. And also now they have been able to do a data index prototyping to easily find and access the biomedical data sets. So you will be more interested in looking at the NCBI portal. There you will find a lot of data sets that are available. I will also now take you to the next example of the data set. That's most popular in the cancer genomics field. It's the Cancer Genome Atlas, in short TCGA. This is also one of the most popular data repository when it comes to the cancer research. It has about more over the next 24 years. TCGA has generated over 2.5 petabytes of genomics, epigenomics, transcriptomics, and proteomics data. The data which has already led to improvement in our ability to diagnose, treat, and prevent cancer will actually remain publicly. This is the most important and most beautiful part of this data set is it's publicly available for anyone. So in research community to use. So if you are interested, you should go to this website www.cancer.gov, and then you can have an open access to this data without paying any fee or any money. And a lot of other tools are also available. So it's if you're starting your career in data science and biology, I think you should definitely look at this portal and try to learn some basic things from there. So and its data is huge. As you can see, it's growing like exponentially. In coming years, it will be really difficult to manage such kind of data, and we need computational tools and methods to analyze this kind of data in future. So even more recently, this new data set that has come up in biology is Human Cell Atlas. It's an effort to map all the cells, what are the functionality of the cells, what kind of genes are expressed in a particular cell. So it's basically focused on single cell level, how how the mapping of human, it's basically mapping the human body at a cellular level. And you can see this very huge data. There are almost 4.5 million cells data available on those cells, and from 33 organs from around now 300, almost more than 300 now, in recently if I checked. And there are around 28 projects that are going around in different labs across the world. So if you are also interested, then you definitely look at this Human Cell Atlas data portal. There you will have access to all the data also freely. You don't have to pay any money for that. So it's multi-omics. Basically, it's not only just genomics. It has different types of omics data that has been stored on different types of cells. So now the second part of this workshop is focused on sequencing technology. So you also heard about some basics in the previous session. So basically, what is nucleotide sequencing? It's a genetic, as we know that genetic information is stored in the nucleotide sequences of the DNA or RNA of an organism. Okay, so sorry for this. So genetic information, as you know, so it has been stored in nucleotide sequences of the DNA or RNA of an organism. Now, the process of determining the correct order of nucleotides is very challenging, and it's also a part of nucleotide sequencing. So basically, we have 4 bases, as you heard, and it's in a given fragment. Now, the nucleotide sequencing has huge applications, ranging from genomic studies to the forensic studies, biology, nowadays, and also in coronavirus and COVID pandemic situation. We also are using different types of sequencing technologies to sequence the SARS-CoV, this virus that has caused COVID-19. And so yeah, so we also have different fields to analyze the function of the genes, and different types of sequencing methods have come up by different scientists. And as you heard, the Sanger sequencing is probably one of the oldest method that was developed in 1970s by scientist Frederick Sanger, and this is basic sequencing pipeline, you can say. It's workflow. So all the methods that are available nowadays also follow this basic strategy. So first is that you extract the DNA from the cell, and then you chop large DNA of molecules into fragmentation, because it's easy to sequence fragments of, instead of sequencing billions of nucleotides together once. In the step B, you do sequencing of those small fragments, and in the next step after these have been, I'm not going into depth, but after these are sequenced, you are again going back to assembly of these small DNA sequences. Remember, the objective is to sequence or to find the correct order of these 4 nucleotides. Though it looks very simple, but it's very challenging computationally. You'll have millions of fragments, and each fragment will be sequenced multiple times. So making assembly out of those small fragments is indeed a challenging task, and a lot of nowadays algorithms are available for that. So thanks to those researchers who have contributed to this. So after this use of different algorithms, you come up finally with the a sequence of 4 nucleotides. So this is our nucleotide sequence. So this basically maps back to the large DNA sequence. So no matter what method we are following, so this is the hallmark of this sequencing approach. And now coming to NGS sequencing. So NGS refers to a modern high throughput sequencing process. Because the systems that are followed here in the NGS are high speed, they are more accurate and cost effective in comparison to Sanger and all traditional other approaches. And the beauty is that the large number of DNA or RNA strands, millions, can be sequenced parallelly, which allows sequencing of the entire, NGS allows sequencing of the entire genome of organisms within a short time period, unlike Sanger sequencing, which takes really days or more time. And the number of different modern sequencing technologies that are available nowadays are basically three. Illumina sequencing, Roche from Roche company for 454 sequencing, ion proton sequencing, and also nowadays solid, which is sequencing by oligo-location detection sequencing. These are different most popular technologies nowadays available in the era of NGS, and they're basically using different approaches for sequencing. So, for example, sequencing by synthesis by Illumina, and sequencing by ligation and ion semiconductor sequencing. These are different methods that are used in. If you're more interested, you should definitely read about those methods if you are planning to make a career in the sequencing technologies. So this workshop, we will not be able to cover all these technologies and methods because of the shortage of the time that has been allotted for this. So just give you an idea why NGS is more important, or why NGS has become so popular in comparison to traditional sequencing methods such as Sanger sequencing. So NGS, as I said, refers to a high throughput sequencing method process. It describes a number of different modern sequencing technologies, whereas Sanger sequences is a sequencing method developed by Frederick Sanger, which determine the precisely nucleotide orders of a given DNA fragment. In terms of cost, the NGS is cheaper process because it reduces time, manpower, and chemicals, whereas Sanger sequencing is costly and it also takes time. In terms of speed, it is very quicker, and nowadays in one day we can do sequencing of whole genome in humans, and it's because this chemical detection and signal detection of many strands are happening in parallel. That makes it faster compared to the Sanger sequences, which is time consuming and it's going serial wise. So NGS is reliable, Sanger sequence is less reliable nowadays. NGS requires less amount of DNA, whereas in Sanger sequence, a large amount of template DNA is required. So amplification has to be done, which requires more, which also makes it technically more expensive. DNA-based per sequences, the number of DNA bases per sequence fragment is lower than in Sanger method, whereas generating sequences are lengthier than NGS because we are doing it in parallel, so fragments are smaller in NGS. So now going next to the field of omics. Slowly, I'm taking in the field of omics. So when it comes to these two different domains, biology and statistics, what everyone now feels that a close interaction between statisticians and bioinformaticians and molecular biologists is essential to provide a meaningful results from any biological problem. So nowadays, unlimited quantity of data are available from from multiple and heterogeneous sources, and there are different challenges, computational challenges, biological issues. Biological interpretation is also a big issue for validation, and this is we need to keep a pace with the new technologies that are coming up. So this cartoon basically just, I like this cartoon, get all the information you can. We will think of use of it is from a biological point of view. And then, yeah, so this is a holistic view of biological system. In nowadays, it's not just cells and biology. And we also have, we have DNA at the level of DNA, RNA, protein, signaling networks. We also nowadays have environment also as an important part of our biological system, because it's always a debate of nature versus nurture. So we believe now that environmental should also be a part. It has been also been proposed by many scientists that environment cannot be separated from our field of biological system. So this is the introduction to different types of omics data. So that is available in biology. So I also introduced in the beginning. So as you see that we start with the transcriptomic data, that is basically from DNA to RNA, how the RNA is transcribed. So we have transcripts at this level. Then we have proteins, how these proteins are translated after transcription. So from RNA to protein, we have a different. If you have followed the last session, that we have now a different level, and we have now in proteomes, we have sequences of protein structures, and how different proteins are functioning, protein interactions. It's kind of data is available at this level. Then we have metabolome level, which are including the chemicals, metabolites that are interacting again back to different levels of the data. And most importantly, though it's nowadays also part of omics, is microbiome. So how the different microbes in your gut are interacting with the host, that is also nowadays considered as a part of omics biological dogma. And now also there are other layers of data in between, though, but if you look at this at a broader scale, we also have nowadays clinical data, such as clinical history of the people, of the person, of the individuals, what are different other records, like ECG and other reports that are available from clinical data, like also molecular profiling, so things like that. Yeah, but omics relations are not straightforward. So that is the message of this slide. So yeah, so as you see that the data is really huge, and what we are like intending, or what is our plan, is to integrate different levels of this information from different levels, and making sense of these different levels of data is not that straightforward. There are several challenges from computational point of view, and also like in this picture, if you see how we cross-check this relation between genome and proteome, and how proteome is related with metabolome and transcriptome. This is just a smaller picture. The overall picture is really huge, including when you're including microbiome, when you're including clinical data in this picture set. Yeah, so one of the interesting goal of this omics data is to integrate different layers of data. So as you see that we have DNA, RNA in genomes, we have transcriptomes here, and we have proteomes, metabolome, and how we can make the conventional approach is that we do a single level analysis. So we do genome analysis on genome level, and then we do transcription analysis, gene expression analysis, things like that at this level, and then we have protein interaction studies, and then we separately also analyze. So this is probably the conventional way of doing this kind of work. But now, when it comes to the single omics and transomics, we can have different directions to go. We can go from genomes to transcriptomics, and transcriptomes and down to different levels, and we can also come back to this to different levels. We can jump to different levels. That is quite challenging, I think, from my point of view, to solve such kind of problems. So we should adopt a holistic view rather than a traditional reductionist approach when it comes to omics data integration. Yeah, so different definitions that we should be familiar when it comes to integration of the data in multi-omics. So let's say we have one side in this slide, we have showing that lipidomic analysis. When we extract the lipids, we do follow this protocol, and what we do is we follow this reductionist approach. We do data analysis on this level, and then we come up with identification and quantification of lipid species that are relevant for the disease, you can say. And other side of this, we can have proteomics analysis. We can do peptide mixture and all. This is all protocols that are followed in proteomics analysis. And it comes to the identification and quantification of proteins that are relevant. Now, more holistic approach will be to that how we integrate this kind of information together. So this is the idea of this omics integration approaches. How we integrate different kinds of information or different types of data that we have got. Data integration from an analytical point of view. So that was in the last slides. You've seen more known technicalities of the omics integration. So here we have, when it comes to analytical point of view, we have different techniques that people should be learning before they get to use to such kind of research work. So for mathematicians, I think these things are probably familiar. So we have matrix factorization, Bayesian approaches, network basis networks, multiple different steps that are available in mathematics. So we have matrix factorization, which kind of separates this information into different factors as latent variables also. And then we have in Bayesian, we have priors, and then we infer the posteriors. And we have in the network-based analysis, we have different networks, and how do we integrate, find sub-networks, and such kind of works is done. And in single omics analysis, we also have information from different levels, and then how we can correlate different information that we have or results that we have obtained from different levels into this together in an integrated way. So finally, we reach to this approach of integration of different methods. How we integrate PCA, matrix factorization, Bayesian approaches, and network-based analysis, and also single level together in this integration paradigm. So what do we expect out of this integration is probably that we want to make an overview of the role of each omics in biological system. So this is our expectation. We want to describe the role of each omics in biological system, and also we want to have a better understanding of the relation between or across the omics types. So molecular signatures with insights in we intend to find molecular signatures with insights into molecular mechanisms. We also have a predictive analytical model that can help us to infer or do some predictions for the patients in future. And probably all of these are our expectations out of this omics data integration approaches. So the idea and the focus should be here: integrate and reduce the dimension of the omics data into a multivariate fashion and identify molecular signatures. That should be our overall focus in this data integration approach. So there will be another talks in the conference also in regard to the multi-omics. One will be from Harvard Medical School, so I definitely recommend you that if you are interested, you should attend that talk from Professor Jessica. This is coming back to omics. So tools. So if you are familiar with some of the tools that are available in online, which are open. So this is one tool that is Mix Omics. This is basically in R. So R is a freely available software package that can be downloaded and used. So different people are using R packages for doing the data science and also. So this is, if you are familiar with the R, then I think I will recommend you that you use Mix Omics. It's one of the most beautiful R toolkit for multivariate data analysis and statistical integration of biological omics data. As you can see, there are different, its focus is on feature selection and visualization. So these are different, this is just an overview of how the work is done by this Mix Omics toolkit. So you have input, which is basically different layers of data, genomics, transcriptomics, metabolomics. And so these are multivariate methods that are incorporated as a part of this tool, like PCA, IPCA, and all PLS. And Tableau is one of the, I think, most popular feature of this tool. And Mint is also another advanced technique to kind of integrate this kind of the multivariate analysis of these different sets of data. You see that it's also different approaches. It's using horizontal integration, it's using vertical integration. So these are different approaches that are followed in these two strategies. And another important, is the graphics. So that is also how you visualize the data. So there are different visualization options also available on this in this R toolkit. So different integration, as I said, is Tableau is following the N integration, Mint is following the P integration, where data sets measure on same P features but from different independent studies. So just skipping this part because I'm running out of time. So yeah, so the take-home messages from this session of the workshop is that we are entering into a new era that first requires data-driven approaches to make sense of big biological data. We are in an exciting era of time, and from a holistic to hypothesis generating approach, we should be thinking in this way if we want to really make, if we want to go and use such kind of data. And as you see that the data does not exclude a priori biological knowledge. We need to have some prior knowledge from biological point of view, maybe the pathway analysis in a computer science method, for example. As we go with the time, the new fields are emerging, and that blends computationalists with biological scientists and vice versa. So we need to have people from different backgrounds to be working in this field to come up with some fruitful results. The development of computational tools is an important part of this field to advance the knowledge. So with this, I would like to thank, and I will end my talk with this beautiful picture that I took from Santiago Lombarda, and this is one of the so what this is showing is the complexity of the data. So it's basically each node over here is a cell, and as you can see, there are a lot of data that has been integrated in the background to cluster different cells together based on different gene expression data, based on how they are different metabolites are probably interacting. So you can see that this is the complexity of the data is so huge that it's challenging, and making sense out of this data is actually interesting and will be challenging for those who would like to make their career in that science and biology. So thank you so much.